I'll be moderating the first session, and let me introduce our first speaker. For many of you, he probably doesn't need much introduction. Dr. Mike Watson got his PhD at the University of Alabama at Birmingham. When I was there, we had what was known as Mike's room, in where he did his work. And you know him now for his work as the executive director of the American College of Medical Genetics. And so we've asked Mike to come talk about this idea of LDTs and FDA regulation. Thanks, Mike.
Is it coming? Okay. All right, so I'm going to really cover three different aspects of this. I'll give you a sense of how we got into this mess with LDTs, talk about what regulators are trying to do to us, and then talk about what we would prefer they did to us, because the plan that they've come up with is pretty much an unworkable regulatory scheme. So I think most people who work in genetics labs know that really about all we've ever had is LDTs in genetics. There's a few more products FDA's approved now for genetic testing in the area of devices and kits, and largely that's because of companion diagnostics. The very few FDA approved products are either at a population level of use or they're products that are used in relatively high volume and mostly in somatic cancer. Very little is available in germline genetic testing. And I think there's a general sense in the community that we do need to improve the oversight and regulation of LDTs. They are a bit freewheeling and they can get out into use a little bit prematurely at times. There's very little control over pre-market release of these new tests and little review of them. So I think everybody's in agreement that we do need to improve the oversight. We're just not convinced that the FDA is the best way to do that. So the two fundamental ways by which laboratory testing is regulated and overseen at the federal and state level is either FDA, that regulates historically the manufacturers of devices and kits, and then the CLIA regulations that really regulate the laboratory practices themselves. So FDA for the products we buy, CLIA for the way we use those products, and now we're stuck in this sort of gray zone where FDA wants to get more control over the clinical laboratory practices. So our major concerns and risks about the proposals that have been made is that there really is little specificity. The two ways you can go about putting new regulations out is either through a rulemaking process, which usually takes 4 to 5 years to accomplish, or you reinterpret prior regulation through what's called guidance. And that's what FDA has been doing, is essentially regulating through guidance by just telling us that now they're going to interpret the way they enforce a law differently than they used to. And rulemaking is supposed to kick in anytime a significant change is made in the way oversight is done. And there's little doubt that an LDT would be FDA regulation of clinical laboratories by considering those laboratories to be manufacturers now would be a significant change. So we've always favored rulemaking as opposed to trying to regulate through guidance. FDA hates rulemaking because they say it takes too long. So they prefer to go through guidance, but I'm not convinced that the current round of guidance isn't going to take as long or longer than it would have taken had they done open rulemaking to establish the regulatory plans really for genetic and genomic testing. A lot of risks. One of the things you get with rulemaking is an economic impact analysis of whatever this new regulation is. And by having bypassed the rulemaking approach and gone straight to guidance, we really have no idea what this would cost laboratories. FDA has a mechanism whereby they typically charge users for the thrill of being regulated by them. Certainly manufacturers are dealt with that way. They pay significant amounts of money to bring their products through. It can be $100,000 or more for what are called PMAs, or pre-market approval applications. It can be $6,000 to $8,000 for the 510Ks, which are sort of predicated on the fact that there are similar tests out there, so they can have a lower kind of regulatory process to run a test through. So we're very much concerned now that this is going to lead to closures of laboratories. I'll try to give you some information on at least one laboratory at the University of North Carolina that's done a cost analysis of what the impact of this guidance would be on their clinical laboratories, and it is far from trivial. So I think there's significant risk of small academic innovative laboratories having to close their doors. Some already have, in fact. So we have this sort of faith-based oversight process that's developing. We have no economic impact analysis done, so we really can't judge whether or not this is going to have a significant impact on access to testing and the laboratories that develop tests. We have very little specificity as to what constitutes high risk, low risk, moderate risk. That is the way FDA regulates. Typically, they call themselves least burdensome and risk-adjusted oversight. And I'll talk a little bit more about some of those risk parameters as we go forward. ACMG has a position on how they would subdivide up different kinds of tests across these different risk categories. We have little knowledge of the cost and no way to really assess how this is going to impact the delivery of genetic testing. I think most people think about molecular diagnostics and some molecular cytogenetics, but it's not just those tests. There's no doubt that FDA is looking hard at newborn screening and regulating state laboratories. There are products used in biochemical genetic testing laboratories, substrates and things that would be considered LDTs because the products aren't regulated by FDA. So the history of this, I had some trepidation when I put these slides together because they actually reflect how old I am. Because I've been watching this go by since I started in genetics back in the late 1970s. That's when the Safe Medical Devices Act was put into place. And one of the reasons why there has been so little regulation of genetics in the Safe Medical Devices Act, they basically grandfathered everything that was being used at the time that law went into place. So all cytogenetics, cell culture, all stains used in cytogenetics, which were the primary type of genetic testing at that time outside of some biochemistry, all of those were grandfathered in, which meant they didn't have to go through any regulatory processes on an ongoing basis. In 1989, things began to change because CLIA went through the Clinical Laboratory Improvement Amendments of 1989, put a significant set of changes into place for the oversight of laboratories. But if you were actually working in labs at those times, you know that CLIA was fundamentally driven by the fact that pathology labs that were doing pap smears and things like that were just running ridiculous numbers of slides through technologists. So fundamentally, they wanted to clean up that part of pathology testing where they felt that there was no control over the amount of volume of work that was being pushed through individual technologists, leading to problems in labs, incorrect determinations of pap smears and that sort of thing. So almost all changes in CLIA were driven by that and actually were focused on that, and everybody else sort of got dragged along with their interest in trying to clean up pap smear testing. But it did open the door to increased oversight, and FDA began to look very carefully at how they might begin to engage with genetic testing at that time. And the first step they took was sort of in the early mid-90s with a category of products called analyte-specific reagents, or ASRs. Those were products that they considered to be a critical component of a test that a laboratory was going to build into an LDT. So these were primers at the time, probes that we might use in molecular diagnostics, and those sort of things. So the next major round of FDA trying to get a handle on genetic testing came with the MAAAs that they began to define in the mid-2000s. These are multi-analyte assays. So basically, these were tests where you tested for a whole lot of different biomarkers. There was a software-written algorithm by which you determined how those various risk factors came together into an overall risk for an individual based on that test. And the information that weighted the different parameters of those kinds of assessments were kept proprietary. So there was no way for a physician to actually confirm the result they were given on these MAAAs. That crashed and burned after about 2 years, and they don't talk about them much anymore. So that was another failed attempt to try to get into genetic testing oversight. And the most recent attempt, I think, is now all around the LDT and the guidances FDA has put out to try to get a handle on it. One of the things that they've put out to justify why they think increased oversight of this kind of testing is required were 20 case examples that they published. And frankly, if you look very carefully at those, what you'll see is that most of the examples of what they called problems based on LDTs were actually things that are not within FDA's purview. Many of them were tests being ordered by people who didn't understand the testing, communicating the results back to patients who didn't understand the interpretation of the test. So if you look very carefully at their case examples, those are not things that FDA actually oversees on the analytical side of testing. So that last major reform is really the late 1980s. But a lot has changed since then. Our testing obviously has gone through a revolution with genomics. There's much increased in the complexity of the testing that's being delivered in genetic testing laboratories. Huge growth in volume since the 90s and early 2000s. And I think the reason that we ended up with this problem is that there's a significant difference between the way FDA oversees drugs. There's a thing called the Orphan Drug Act that actually has worked well to incentivize manufacturers to develop products for rare diseases. They get a period of monopoly of the particular drug they develop where nobody can compete with them. They get tax incentives. And that really did drive industry to more actively begin to develop the kind of drugs required for genetic diseases and rare diseases. Unfortunately, we don't have that on the devices side of FDA. The humanitarian device exemption really has never worked. It's never incentivized manufacturers to bring these kind of products forward. The clinical trial costs were far more expensive for bringing a product to FDA when your return on investment for diagnosis in a rare disease was going to never actually recover your costs of developing that test. So manufacturers wisely said, we're just not going to go there, left the laboratories to actually have to develop the test. They knew that the capabilities were there and the products were available that we could put the test together ourselves. And that's why we have this LDT world. And to this day, there really isn't much in the way of useful incentives. I think we're in this major transition in the kind of testing that we'll talk about, and David Bick will give you more on genomics and exomes and things later in the meeting. But it's certainly not an area that they have much expertise in overseeing. So commercial kits versus LDTs are certainly not the same. The LDTs that we use in our laboratories typically are used on a local population. Kits and devices that manufacturers make are purchased by laboratories and typically delivered to a much broader segment of the population. And if the tests are good, the laboratories do buy them. There's lots of test devices and kits that have been used in laboratories. And when they go through this long process of development and oversight, they are typically good products and are easier to use than would be the laboratory developed test. Unfortunately, we don't have many of those to work with at all. So LDT design, development, validation is all very much tied to the performance of the test. Commercial kits tend to be engineered for standard patient and standard laboratory types of services. Excuse me. So we're trying to figure out what is this right balance to strike between sort of an FDA role in laboratory genetic testing versus the CLIA oversight of the way laboratories deliver testing services. There's a number of aspects covered by CLIA. It certainly covers quality assurance, quality control, sort of your reference ranges and all those sorts of things, the personnel that do the testing. And it's only been this product side of manufacturers that has been under FDA and with these sort of mass-produced and manufactured commercial kits. So now to give you a sense of what the implications of at least the guidance that FDA has put out would be on clinical laboratories. Jim Evans at the University of North Carolina did an analysis within their clinical laboratory system of what the costs of complying with this guidance would be for them. So within their core laboratory, including chemistry, hematology, coag, cytogenetics, molecular pathology, and molecular microbiology, they identified 258 LDTs that were being delivered by that laboratory. LDTs in this context meaning a test they've completely built from scratch. However, there's another group of tests that are also captured under this, which is any test that FDA might have approved as a kit or a device that a laboratory buys and changes in any way, makes it an LDT. You have to go back through your own validation processes to really show that that test is performing the way it's expected. Whereas with just a kit or FDA-approved kit or device, the responsibility is to show that your delivery of the test, your validation of the test, meets that basic validation criteria that came in the packaging materials with the test. So you have to show that you can run the test at the same level that device had been approved at, as opposed to independently going in and validating a test that you've created yourself. So minimal cost for complying with FDA oversight will have to include registration. So right now we're in this process where every laboratory is registering. I actually watched when I co-chaired the task force on genetic testing for the Department of Energy and NIH back in the mid-1990s. Steve Gutman from FDA was a member of the task force, and he laid out he had showed us a plan at that time that FDA had developed. It was 25 years long, and this was their plan for getting a handle on genetic testing. The first 2 years of the plan were basically, who are these people and what are they doing? They didn't even know where the genetic testing laboratories were. Their CLIA captures this from anybody who runs a lab knows that when you get your CLIA license, you send them a list of all the tests that your lab's doing, and you're expected to keep that list up to date, but nobody really did. When they added a new test, they never told CLIA. CLIA did all this on paper, so FDA had nowhere to go to try to find an electronic record of where all the clinical testing laboratories were that were licensed by CLIA and what they were doing. So this first level of registration for a couple of years was really just to figure out who they are and what kind of tests are they doing. And then they would begin to roll out this sort of risk-focused oversight program, starting with what they thought would be the highest risk tests. All right, I said all that. So if you begin to look at sort of the assessment of what it costs just for these registration processes, it's providing a lot of data about the validation, your analytical validation, and a lot of clinical validation data. They estimated that it would cost them about $2,000 per test to comply with the registration process. So based on those 250 or so LDTs that were being delivered, they were looking at about $1 to $3 million each time you went through the registration process, because it was going to be a recurring process. All right, so what are your trade-offs? So if you're doing a 510K, which is what once we get through this first wave of registration, where the tests that are currently in practice would be not really grandfathered like had been done in the late 1970s, but would have a lower level of oversight associated with them. It would be, after those have been approved, there's then going to be a process whereby you apply on a test-by-test basis as a new test comes online. And if there are other tests like that test out there, you do a 510K. And that's going to cost between $6,000 and $7,000 or $8,000 per test that one would submit to FDA. So it's clear that between registration, notification, the University of North Carolina estimated that they would have to take a very, very significant proportion of their tests out of their laboratory and essentially have those tests sent to LabCorp. Much of the data we have about how they're approaching this oversight has really been very much driven by the devices manufacturers and by the very large clinical laboratories, Quest and LabCorp. They can absorb the cost of these kind of oversight things and capture market share as they're doing that. And as the other laboratories that can't afford to comply, begin to close down, then I have no doubt that we'd see what we always see, which is you capture the market and then the prices start going up. So if you ignore the fact that the test costs will inevitably increase over time, the proposed legislation is enacted, reimbursement never keeps pace with the increasing charges, and profits tend to begin to drop in the laboratory sector. The very high fees for regulatory compliance would tend to drive the field towards these sort of very big national reference laboratory systems, and they're essentially all oligopolies and monopolies. Excuse me, I knew I always go dry when I talked. So we've gone down this road before of monopolies in laboratory testing. Certainly Myriad's BRCA monopolies led to interesting problems in the delivery of testing, though those are now essentially gone and been converted now into trade secrets, at least from Myriad's perspective. So we have now this sort of proposed regulatory system that is really, I think, a threat to genetic testing laboratories in the country. So when you think about the FDA typically pays much attention to analytical validity, but their history has been that you only have to worry about analytical validity when you have a test that's clinically valid. So that was the means by which they got into assessing the clinical validity, without which they wouldn't even have to worry about analytical validity. But much of the clinical validity aspects of genetic testing are sort of embedded into the practice of medicine and are much less amenable to the sort of the regulatory oversight approach. They're not under the exclusive purview of the clinical laboratory. Most of these LDTs are tests that result from interactions between laboratories and the medical literature and the providers in an institution who want tests that are not typically available to them in the laboratory marketplace. It's unrealistic, I think, to imagine that FDA could actually adjudicate clinical validity. There's like 12 people. And we're talking about an enormous number of tests right now. We're testing somewhere in the neighborhood of 3,500 genes right now. And FDA, I'm pretty sure, doesn't have anybody board certified in clinical genetics, medical genetics, working on their staff. They have some folks trained in molecular biology. And their history, frankly, of overseeing the manufacturers led to a very warped view. They frankly don't understand. They rarely got into clinical laboratories, so I don't think they understood what they were proposing to regulate within clinical laboratories. In fact, thousands of clinical laboratories all across the country. And to a large extent, clinical validity can be assessed and quantified via a systematic survey of medical literature. We're doing a lot of work around clinical validity in ClinGen right now, and I think what you're going to begin to see, in fact, I'll mention towards the end a new guidance that came out, I guess it was Friday of last week, on next-gen sequencing. The FDA put out 2 guidances of how they intend to regulate those. One regulates the clinical validity side, and one is a guidance about the analytical validity. And it's clear that they're going to have to rely on large databases. The days of being able to run large for every laboratory to run a clinical trial are gone. It doesn't work in rare diseases, and really what we're going to be having to deal with is establishing clinical validity with data sharing through things like ClinGen. FDA's first approval of the Illumina sequencing platforms used the CFTR2 database that was built by Gary Cutting at Johns Hopkins. It's an enormously well-resourced activity, the CFTR2 database. The Cystic Fibrosis Foundation throws a ton of money into it. They throw a ton of money into centers across the country that follow the diagnose and manage these patients, heavily subsidized. So they were able to build a system, and everybody thinks it would be wonderful to apply that to every genetic test we do, but the reality is only the CF Foundation has $3 billion that it got from the development of the Vertex drugs for CF that were targeting specific mutation types. Most other diseases, Muscular Dystrophy Association can put some money in and deal with those, but the vast majority of genetic tests just aren't going to be able to take advantage of what cystic fibrosis was able to do. And now we're building these sort of public databases like ClinGen that will establish clinical validity, and not just in the typical way of that there's a clear clinical association between a particular biomarker and a phenotype or disease. It's one of these processes that's changing, excuse me. That's changing quite rapidly. It's not a yes-no association anymore between a particular gene and a phenotype. It's a question of how good is that association. And frankly, as we begin to standardize the processes by which we look at validity, it's clear that there are conflicting claims on some variants in genes. There are some associations with genes and phenotypes that are very strong, definitive associations, all the way down through weak and conflicting. So it's clear that the way we use different kinds of genes in tests is going to have to change when we build panels of tests. In fact, I think if you look at there's a paper in Genetics in Medicine that just came out this month on hearing loss, and it talks about looking at, and obviously these are patients mostly identified in newborn screening programs nowadays. The Odo chip was a test that was delivered by the molecular lab at Harvard Partners, had about 160 or 70 genes on it, and these were really anything where a particular gene had been identified as having an abnormality in a patient who also had hearing loss. When they began to really apply some strong standards to looking at these associations, I believe it was in the neighborhood of 50%, 45 to 50% of genes would be taken off of these kind of panels. And as you move into screening kind of applications, then I think you need to get even more definitive about your knowledge of these associations. So these intended uses are really going to mean that any one test could actually be a whole lot of different tests depending upon whether you're in a diagnostic setting, a screening setting, and such. So that's going to be one of the challenges in this LDT world, is that it's it's the intended use of the test that determines how many tests there really are, not the fact that you have a test that has a very specific set of analytes in it. All right, so FDA's current approach we don't think is very workable. Any laboratory test can be a high-risk test, depending on certain clinical contexts in which that test might be done. So the whole risk categorization scheme is a very complex one to try to apply uniquely to genetic tests. Not a particularly good parameter to calibrate how you're going to oversee different types of tests. To prevent the problems from the test with no clinical validity, regulatory agents have to focus on the claims the laboratory makes as to what that test can actually do. And these can be quite dramatic claims. Now, I'm going to zip through this one because I've said most of what's on it. So, one of our major concerns is, can FDA actually oversee genetic testing or not? As I've said, I don't think they can. They certainly don't have the staffing. They don't have the clinical backgrounds that allow them to get heavily into genetic testing. The LDT draft guidance that came out is finalized in essentially the same form that it came out before. FDA had told us early this year that when their next guidance came out, that was it. There was going to be no changes, and they've been saying it's coming out in about a month for about 6 months now. There's a ton of congressional activity taking place in the backdrop because there are many in Congress who are worried that FDA really has overreached in trying to take on the laboratory-developed testing area. If you think about these sort of risk classifications that FDA operates on, they're obviously low risk, moderate risk, and high risk tests. The low risk tests are typically ones where the consequence of an incorrect result or an incorrect interpretation is unlikely to lead to serious morbidity or mortality in the individual to whom the test has been delivered. The moderate risk, and there's very few of those, frankly, in genetics. The great majority of what we do would be in this moderate risk category, where the consequences of the incorrect result or interpretation could lead to serious morbidity or mortality. But there are physicians in between the laboratory test and the patient adjudicating whether or not the risks of treating or intervening in that patient based on the test outweigh the risks of the disease itself. And then there are the high-risk tests, which in our mind are those algorithmic tests for which there is no way to confirm the result of the test. You don't know how they weighted the different parameters in coming up with an overall risk to a patient of having a particular disease develop or having a particular disease evolve in them. Again, in the low-risk tests, the test result is typically used in conjunction with other clinical findings. The moderate risk test is typically for diagnosis, predicting disease progression, identifying whether patients are eligible for specific therapies or not. And then this whole area of the high-risk test, where the test result predicts risk progression for a patient's eligibility for a specific therapy and uses proprietary algorithms. So there's a lot of groups that have been working together. We work closely in a coalition that the AMA has been umbrellaing, CAP, the Association of Molecular Pathologists, Infectious Disease Society of America. There's a lot of different groups that have been participating in trying to find an alternative approach to the LDT guidance that FDA put out. And we're really looking at a much more of a hybrid model, which requires significant enhancement of the CLIA regulations to improve laboratory practices associated with genetic tests. Now, in this aspect, or in this approach to modifying or enhancing CLIA, you can see that there are 3 categories of testing under CLIA. There are low-risk tests, moderate, and high-complexity tests, and that's fundamentally how CLIA is structured, is around how complex a particular test is. So we would leave everything in the low and moderate risk categories of CLIA as they are. But in this area of LDTs, we would subdivide it up into 3 categories based on those risk parameters that are similar to the way FDA thinks about it and would be closer aligned to ACMG, CAP, and AMP's positions on how one divides up these risks across different kinds of tests. And that whole subset then would be regulated under CLIA, but only the high-risk tests would go to FDA. We do think that if a test is high risk, it does need a high level of oversight. And if the manufacturers or the people who are developing those tests are unwilling to tell you the parameters under them, it's very hard for a group to assess them independently. And FDA does have mechanisms for confidentiality and getting proprietary data and all that kind of thing. So we felt that FDA ought to be dealing with the high-risk tests unless the manufacturer is willing to be transparent about all of those algorithmic components of the test so that everybody could see exactly how they go about weighing all the parameters. All right, so a lot of activity in Congress right now. This is going to be in the House of Representatives. The Energy and Commerce Committee is the body that deals with FDA and deals with the LDT issues. They have a bill that's called the 21st Century Cures Act. It had a little bit of LDT information in it, but it's been gutted completely because there's no consensus in Congress as to how to approach LDTs. Well, let me back up. It's actually been one of the more bizarre processes I've ever actually been involved in watching the House Energy and Commerce Committee. They organized a meeting back in January, and what was required of anybody that came to the meeting was that they agree with the approach that they were beginning to take on regulating LDTs. So we obviously weren't even able to go to the meeting because we didn't agree with the approach. So by the time the meeting was over, everybody agreed with this proposal that Energy and Commerce, and I think they realized that the process was so flawed that it's just been stuck in Congress now for almost a half a year. On the Senate side, the Senate Help Committee is the place where much of this LDT activity is being dealt with. They're approaching it quite differently than the House Energy and Commerce Committee has. Timelines are really unclear. We know that the goal was to get the LDT language into legislation so that they could either support or fend off FDA's approach to LDT regulation. Unfortunately, they go on their summer vacation, I think it's the end of this week, and there's nothing yet on LDTs in any of the Senate or House legislation. So I mentioned that there was something new out that came out just on Friday of last week, FDA's approach to next-gen sequencing. And it's two different guidances. One is very much targeted at analytical validity. It's called the use of standards and FDA regulatory oversight of NGS-based in vitro diagnostics used for germline diseases. So they considered germline to be easier than somatic and targeted it as their first set of guidance. But there's an underlying issue, which is that almost all tests we use on next-gen sequencing are LDTs. So nobody's going to be required to actually comply with the next-gen sequencing guidance until the LDT issue is dealt with, because so many of the tests are LDTs. The second guidance is really clinical validity, and they're arguing that FDA is going to begin to oversee and approve extant databases like ClinGen or like CFTR2 as tools that laboratories can use to document clinical validity. And they're actually setting up a paradigm whereby if a manufacturer wants to keep their information secret, then they will have to go through the full PMA or 510K process to get a new test approved through FDA. However, a laboratory, or anybody actually, a manufacturer could do it as well, that's willing to base their clinical validity claims on the data that sits in these extant databases would be the approved process for clinical validity. And they're suggesting that anybody who would use that would be exempted, could be exempted in the long term from having to even apply to FDA for approval of their test. So we're at the stage where we're organizing another webinar for September. We did one back, I think it was in March, trying to engage the community and get them to start talking to their representatives about the implications for this kind of oversight on their laboratories. The cost issues in and of themselves really gave pause to many laboratory directors, and I think the barrage of comments that went in back in March really slowed down the juggernaut that was developing in legislation. We obviously favor the CLIA-centric approach. CLIA can be enhanced. What we are proposing is that there be a mechanism, something in the third-party review systems outside of regulation. If FDA was to do this, they'd have to bring all the experts together on panels. The alternative is for the professional community to deliver a service much like we do inspections and laboratory proficiency testing, offer a new type of service whereby a group could review new tests going online and approach them in a much more fiscally responsible way. I can't see every laboratory in the country having to convince somebody that the same test is valid. So I can see developing a mechanism whereby once 5 labs have shown you the same test, there is no longer a need for every laboratory to have to go in and do that same validation study. So there's a lot of things that we can do in the private sector that aren't part of what the regulators have been thinking about. And so on that I'll stop. I see I have that 1-minute sign pointing at me. So I'm happy to take questions on LDTs or the new next-gen sequencing guidance that just came out. And you'll certainly, I know that I'll make the regional collaboratives aware of the webinar that we'll do in September to try to get people up to speed. There's a 90-day comment period that'll end in around the 1st of October on the new guidances, and we do want people to get up to speed on understanding the implications so that they can make their own case to their own representatives. Questions? Yep.
So none of this obviously makes sense, as you pointed out. There's this overlap between what's being proposed and what CAP already does, and you didn't really mention very much about CAP proficiency and oversight of laboratories in general, but wouldn't this fall under the purview of CAP proficiency testing and that sort of a, so is CAP talking to FDA or?
Well, proficiency testing is under CLIA. It's really not an FDA. The only place FDA gets into CLIA-related things is in helping them determine what is high complexity, moderate, or low complexity. They have a behind-the-scenes partnership.
But CAP runs the proficiency testing.
They do run the proficiency test, but they do it as a deemed body under CLIA. Your licensing is because they're deemed to deliver your license because their standard is equal to or higher than that of a federal standard. Some states are exempted because their standard's higher. So yes, CAP does do licensing and it does do proficiency testing. And we've had, for 2 years, we've been talking about building this new piece under the CAP system because they do connect to all laboratories that would be the third-party review process for adjudicating validity of new tests that are going to be offered.
But I guess what I'm asking about, has FDA just blinded themselves to that whole process, or it seems to me there's redundancy between what you're proposing and what's already existing.
Yeah, no, there is. Right now, there is, what FDA is proposing, there is a lot of duplicate stuff that is very similar to what's under CLIA. And all of our recommendations back to Congress and others have argued that we have to separate these pieces out. We can't be having to duplicate these activities under 2 different regulatory structures. So getting harmonization of CLIA is part of, with FDA, is part of that enhancement process.
Do you have a bullet point document that we can find on your website to use with our legislators?
On LDTs?
Yeah.
There are certainly our letters are on our websites to various
Which website
bodies.
Specifically?
Excuse me?
Which website? Just so we can all go there and look at them.
So on the ACMG site, there's a bar, on the toolbar, there's an area called Advocacy. Under Advocacy, you can find the LDT
Thank you.
materials. We did a webinar in March, AMA, Jim Evans talking about the implications at UNC. All those are available on that Advocacy tab as well. David.
Hi, I'm David Bick from Hudson Alpha. So logically,
that's a bad place to start.
I know, it's a bad place to start. So logically, we would have a body basically go through each of the 20,000 genes and make an approve, disapprove for each gene. But then in addition, you'd probably, I guess, want to have like off target because sometimes genes have more than one disease.
Well, yeah, I'm not worried about all 20,000 just yet. I mostly will worry about the 3,500 or so that actually have tests associated with them. But yeah, it's still a very high volume of testing. What was the second part?
Oh, just that, once you approve a gene, then
Oh, yeah,
you would have to then go back periodically and approve the new use of that gene because someone found a new phenotype. And then you'd
Yep.
have to say that, well, maybe CF has 3 phenotypes, or maybe it has 4 phenotypes.
Yeah, yeah. No, honestly, it's an absolute mess. I mean, if you look at the FDA LDT guidance, every time you would add a new variant to a particular test, you would actually have to go through this entire process again.
Got it.
They're not allowing you to just acknowledge some very incremental improvement in clinical sensitivity with the new variant. It's a brand new test, and it comes back through the process. So it really does inhibit innovation tremendously.
Right, so every variant at the end of the day gets approved. Got it. Okay.
Yeah, no, I think everybody, that's why we have LDTs, is that once you get FDA approval, it locks the test down. And they're locked down for a long time, and there's no incentive to improve them because of the costs involved in going through the process.